Hi guys, today we're going to be looking at DNA and RNA, our nucleic acids. DNA or deoxyribonucleic acid is found in all living things. If it's inside of a nucleus, that's a eukaryotic cell. But if it's just floating free in the cytoplasm, that's a prokaryotic cell. So, what's so important about DNA? Well, DNA is the recipe book for proteins, and it controls all of the processes that take place in your cell. And so, DNA will be turned into or read or copied into mRNA, and that mRNA will be read so that we can make a protein. And the proteins are how we express our genetics. So, by reading our DNA, we can express the traits that are needed to run the cell. And every cell will read different parts of the DNA so that it can accomplish what it needs to do. So, let's look at the structure of DNA. It's made out of these nucleotides composed of a phosphate, a sugar, and a base. The sugar for DNA is deoxyribose, and the bases for DNA are A, G, C, and T. Now, the structure of DNA, these nucleotides are paired up on the left as well as on the right. You'll notice that the right side is upside down, and that is on purpose because DNA runs in opposite directions, which means it's anti-parallel. You'll also notice that A adenine always pairs with T thymine with two hydrogen bonds in between them. So we say apples go in trees. And then you'll notice up here C with G with three hydrogen bonds between them. Say cars go in the garage. So you'll always see those paired up together in DNA with the hydrogen bonds between them, which is important because hydrogen bonds are weak and can be easily broken so that DNA can be both replicated and transcribed. So, if you were to be given a side of DNA, to build the other side, you're just going to use the base pairs. So, I'll show you a couple examples. If A is on one side, you put T on the other. G goes with C, C goes with G, T goes with A. So, again, apples in the tree, cars in the garage. Now, you may be asked a question about the structure of DNA and how it's anti-parallel. So, again, anti-parallel just means that the DNA is going in opposite directions. And the biggest type question that you might be presented with on a state test would be something like this. If they give you a little segment of DNA and they put a 5 prime on one end and a 3 prime on the other end, the only thing that you really have to understand is that the complementary side will run 3 prime to 5 prime. And so, what are those 3 and those 5s? What do they refer to? They actually refer to the sugar that's facing up on in the nucleotide. So in this on this side, the 5 prime or the 5 sugar is pointing up, and on this side, the 3 prime sugar is pointing up. That shows that they're upside down. So again, the only time that that might come for you to have to focus on that is if they give you a segment of DNA and they tell you that one side is 5 and the other is 3, the other side has to be opposite. All right, so DNA is twisted up into a double helix. So we take that ladder structure and we twist it up into a double helix. And one last thing that I'll say is that there is a law called Chargaff's rule that states that the percent of A should be equal to the percent of T, and the percent of G and C should also be equal. So if the DNA has 15% adenine, it should have about 15% thymine as well, because they always pair up. So, what happens when we do mitosis or meiosis and we need to replicate our DNA? DNA replication is going to create two identical strands of DNA so that it can be divided amongst the new cells. So, the first step is to open up the DNA to unwind it by breaking those hydrogen bonds. After that, we're going to use both sides as a template and we're going to begin to add new bases onto each side. So we'll add them to the top strand, we add them to the bottom strand. And that's going to start to create two strands of DNA. What you'll notice is at the end, both sides will have an old strand and a new strand. So if you look in this picture, you see it has a dark blue strand and it has a light blue strand. The dark blue being the old the light blue being the new. And so we call DNA replication semi-conservative, meaning it keeps part of the old and part of the new. But the end result is that one DNA turns into two identical copies of DNA through DNA replication. All right, the last thing we need to look at is RNA. RNA is a nucleic acid just like DNA, but there are some differences. So, first of all, the nucleotides that make RNA have a ribose sugar instead of a deoxyribose sugar. That's why it's called ribonucleic acid, because of the ribose sugar. The bases are the same, except thymine in DNA has been replaced by uracil. So, you will not see a T if you're building mRNA, tRNA, or rRNA. They don't use thymine, they use uracil. RNA is also single stranded, so it is a helix, but only with one strand. And lastly, there are three types of RNA. mRNA is a copy of the original DNA, so just a small portion. And it's small enough that it can leave the nucleus, so it can take the message of DNA out of the nucleus. rRNA, this one pictured here, makes up the ribosome. And the last picture, tRNA, this is the transfer RNA, which brings amino acids to the ribosomes, meaning that when the ribosome is building the protein, it's going to pick up the correct amino acid and bring it to the ribosome. The ribosome is doing the building of the protein. So this is kind of like the sous chef that brings it what it needs. All right, that's it for the structure of DNA, DNA replication or copying DNA, and RNA.